Assalamualaikum, welcome back to my video. So in this video we are discussing on cofactor. So enzymes may work together with a non-protein chemical component for enzymatic activity. It is used to increase the catalytic activity of enzyme or act as helper that required for enzyme function. Because cofactor are more stable in higher temperature, and the binding of cofactor to the enzyme can be permanently or reversibly, and they can be used repeatably. Alright, so there are three important types of cofactor, which are the coenzyme, prostatic group, and metal ions. Let's talk about coenzyme first. So coenzymes are organic cofactor. It is dialysable, which means they can pass through the dialysing membrane, and they are thermostable, which means they are stable in higher temperature, and it loosely attached to the enzyme. What are the functions of coenzyme? It makes up a part of the active site of enzyme and prepare the active site for catalytic activity. Since without coenzyme, enzyme will not function well. For example, like this diagram, without cofactor attached to the enzyme, the protein, aka the enzyme, will not active, and with the presence of cofactor, so enzyme can bind to the substrate. Right? Example of coenzyme is NAD. The full name of NAD is nicotinamide adenine dinucleotide, and it is used in cellular respiration. Next is prostatic group. Prostatic group is organic cofactor. It is dialysable, thermostable, just like coenzyme, and it attached firmly to the enzyme. For example of prostatic group is FAD. The full name of FAD is flavin adenine dinucleotide, and it is also used in cellular respiration. And lastly is metal ions. Metal ions are inorganic cofactor. Example of metal ions are K+, Zn2+, Mg2+, CO2+, Fe2+, and Mn2+. What is the use of metal ions? It is used to alter the shape of enzyme so that the binding of substrate more easily. Alright, these are the terms that used in enzyme components, which is holoenzyme. It means that it is the enzyme that has cofactor. Alright, and apoenzyme is enzyme without cofactor. Just look at the diagram. Apoenzyme, which is enzyme without cofactor, at either coenzyme, prostatic group, or metal ion. The protein portion means that the enzyme, okay, apoenzyme, inactive enzyme, and with the presence of cofactor, the enzyme becomes active, and we call it as holoenzyme. So let's summarize. There are three important groups of cofactor: coenzyme, prostatic group, and metal ions. And then they can be divided into two types, which is organic and inorganic. The binding of cofactor may be permanently or reversibly. And look at the example of each cofactor. Coenzyme, many coenzymes are obtained from vitamin. Prostatic group, the cytochrome oxidase has prostatic group heme, while metal ions, you can see that the Zn2+ are the cofactor for carbonic anhydrase enzyme, while the Cl- it is the cofactor for salivary amylase.